Hello Medicose, welcome to the channel. In this video we'll break down the structure of heart muscle cells and explain the step-by-step process of how these cells contract, fueled by the powerful interaction between calcium and muscle proteins. From the Sarcomeres structure to how calcium triggers contractions, get ready to explore the science behind each heartbeat. Myocardial cells are the building blocks of the myocardium, the muscular middle layer of the heart wall. They contract rhythmically and synchronously, propelling blood throughout the body. These cells are characterized by the presence of specialized units called sarcomeres, the basic contractile units of muscle tissue. Sarcomeres are arranged end-to-end, forming long cylindrical structures known as myofibrils, which run the length of the myocardial cell. Each sarcomere is composed of two types of protein filaments, actin, which are the thin filaments, and myosin, the thick filaments. These filaments are arranged in a specific overlapping pattern. The actin filaments are anchored to the Z-lines which form the boundaries of each sarcomere. The myosin filaments are located in the center of the sarcomere and are interspersed between the actin filaments. This precise arrangement of contractile proteins within sarcomeres is crucial for muscle contraction. Myocardial cells are interconnected by specialized structures called intercalated discs. These discs are located at the ends of the cells and appear as dark bands under a microscope. Intercalated discs play a vital role in maintaining the structural integrity of the heart muscle and ensuring coordinated contraction of myocardial cells. Intercalated discs contain two main types of junctions, desmosomes and gap junctions. Desmosomes act like spot welds, anchoring adjacent cells together and providing mechanical strength to the heart muscle. Gap junctions, on the other hand, are channels that allow for the rapid passage of ions and small molecules between neighboring cells. This electrical coupling enables the heart to contract as a single coordinated unit, ensuring efficient blood pumping. Myocardial cells are highly metabolically active and require a constant supply of energy to fuel their continuous contractions. To meet this energy demand, these cells are packed with mitochondria, the powerhouses of the cell. Mitochondria are responsible for producing ATP or adenosine triphosphate, the primary energy currency of cells, through cellular respiration. In addition to mitochondria, myocardial cells also possess an extensive network of transverse tubules, or T-tubules. These invaginations of the cell membrane penetrate deep into the cytoplasm, bringing the extracellular fluid closer to the sarcoplasmic reticulum, a specialized network of membranes involved in calcium storage and release. This close proximity facilitates the rapid and efficient delivery of excitation signals and calcium ions throughout the cell, ensuring synchronized contraction. Calcium ions play a pivotal role in muscle contraction, acting as the molecular messengers that connect excitation to contraction. When calcium is released from the sarcoplasmic reticulum, it binds to a protein called troponin, which is located on the actin filaments. This binding causes a conformational change in the troponin-tropomyosin complex, exposing the myosin binding sites on the actin filaments. With the myosin binding sites exposed, the myosin heads can now bind to the actin filaments, forming cross bridges. This binding triggers the release of energy stored in the myosin heads, causing them to pivot and pull the actin filaments towards the center of the sarcomere. This sliding of the filaments results in muscle shortening and contraction. The process of muscle contraction is best explained by the sliding filament model, which describes how the actin and myosin filaments slide past each other, resulting in muscle shortening. This model was proposed independently by two groups of scientists in 1954 and has since become the cornerstone of our understanding of muscle contraction. According to the sliding filament model, muscle contraction occurs in a cyclical manner, with each cycle consisting of four main steps: attachment, power stroke, detachment, and reactivation. This cycle repeats as long as calcium ions are present and ATP is available, allowing the muscle to generate force and shorten. Numerous factors can influence myocardial contractility, either enhancing or reducing the heart's pumping power. These factors can be broadly categorized as positive inotropic agents, which increase contractility, and negative inotropic agents, which decrease contractility. Positive inotropic agents include hormones like adrenaline and noradrenaline, which increase heart rate and contractility, as well as drugs like digoxin, which enhances calcium availability within myocardial cells. Negative inotropic agents, on the other hand, include conditions like acidosis, low blood pH and hypoxia, low oxygen levels, as well as certain medications like beta blockers, which block the effects of adrenaline and noradrenaline on the heart. In conclusion, myocardial cells are highly specialized cells that form the muscular foundation of the heart, tirelessly driving the circulatory system that sustains us. Their intricate structure, with Sarcomeres as the fundamental units of contraction, allows for the coordinated shortening and relaxation that propels blood throughout the body.